So yes, I will be trying to answer the same question that I tried to answer for the last 18 years since I started my lab. How do you make and maintain a self-renewing human hematopoietic stem cell? When I started working with the HSCs in Sweden in '98, we were let's just find the HSC cytokine so we can put it in culture and expand them. And those who are still looking for that cytokine might be looking for a long time, because we've understood that this is a much more complex process. And actually, it makes sense that regulation of self-renewal has to be complex, because HSCs has to be ready to respond to various needs of the body and in many ways change their fate decisions, at least in part, in a reversible fashion. And this is the only way how we can keep on keeping a healthy blood stem cell pool throughout our life and make all the lineages as needed. And we all know that when you put HSCs in culture, it's often a one-way street towards differentiation or death, or at least those HSC lookalikes, they don't engraft very well. At the same time, we know if we play with self-renewal, we're very easily at the kind of the intersection of self-renewal or leukemic transformations. We have to be very carefully how we use this weapon. And finally, this question, this dream that we've all had, could we use human ES cells or iPS cells to make patient-specific or at least perfectly HLA-matched HSCs for therapies? But now we know that HSC development is a very complicated process, so it's most likely not going to be easy to make this happen in culture. So if we take first a look at HSC development, as Len already alluded, hematopoiesis during development happens in waves, and the HSCs are generated in the last wave. So the embryo is first very busy in trying to keep it alive by making red blood cells and these macrophages that are dooming and grooming already at the time when the HSCs are being formed. So most of these findings, how we view developmental hematopoiesis, have been done in the mouse. But if we look at mouse versus human developmental hematopoiesis, the basic principles that this is divided to 3 waves, where first you make the progenitors starting in the yolk sac, and then ultimately you make the HSCs in the AGM region. This is conserved, but if you look at the extraembryonic tissues, look completely different. So how do we know if this is conserved? Also, the length of pregnancy, mouse is 3 weeks, human is 9 months, so it's easy to understand that kind of the requirements for the different progenitors are different. But also, another thing that many people don't even think about is if we really match the developmental stages, when the mouse is being born, we are only at 9 weeks of human development. So when the mouse is running around already, the human is still being incubated in the womb much longer. So human fetal liver hematopoiesis happens mouse when it's already out of the womb. So it's important, I think, to really, if we want to understand human HSC development, to study this in human rather than just try to extrapolate solely from the mouse. So we know we've learned a lot of important components of this process from our previous studies, key factors that regulate hematopoietic specification, that send the mesoderm towards the definitive HSC fate rather than to those progenitor fates. We found some key markers how we can identify HSCs during human fetal development, such as GPI-80, and this allowed us to get a closer look of the self-renewing HSC. So we've learned these pieces, but we haven't been able to connect the dots to really recreate this process in culture. We know that this involves multiple tissues, multiple organs, but how exactly is this happening? So in one way, we had to just wait for technologies to develop, and thank God, the single-cell RNA sequencing was invented, and we were able to collaborate with our long-term collaborators, Katja Schenkelelen lab in Tübingen, who had access to very early human developmental hematopoietic tissues at the stage when HSCs are being born. So we decided to take on a kind of a comprehensive single-cell RNA-seq study so that we could finally ask, how are the nascent HSCs different from those progenitors that are generated in those HSC-independent waves? And what are the tissues that are involved in this process? We know that HSCs emerge, at least some of them, if not all of them, in the AGM. But there's been hints, at least in the mouse, that the extraembryonic tissues, the yolk sac and placenta, may also contain HSCs. Then they go to the liver, and there in this black box, something happens that they magically learn all the skills that they need to do their adult jobs, and ultimately they go to bone marrow to maintain hematopoiesis. How do these different tissues contribute to this process? And then last but not least, what makes the HSC-forming hemogenic endothelium unique compared to the other type of hemogenic endothelium that makes the progenitors? Because we believe that it's already in this mesosperm specification stage when you're setting the fate whether you are making an HSC or something else. We first looked into the AGM, and based on what we knew from our other studies of key HSC transcription factors, RUNX, HOXA9, MLT3, MECOM, and HLF, we found the cluster that really must represent the nascent HSCs in the AGM. From here, we learned a signature that helps us identify the nascent HSCs compared to the progenitors and other cells in the AGM, and we were able to narrow this to a 6-gene signature that we showed that when we were at any stage of human hematopoietic ontogeny, we can distinguish HSCs from progenitors by using this 6-gene signature. And we were able to show that at the time when HSCs are in the AGM region, we can also find them in these extraembryonic yolk sac and placenta before they have seeded the liver, suggesting that the yolk sac and placenta are important component of human HSC developmental path. And by following these 6 key HSC genes, we could show that HSC transition from the AGM through the extraembryonic tissues to the fetal liver between 5 and 6 weeks of development. 6 weeks onwards, we can find something that is transcriptomically an HSC, but only from 8 or 9 weeks onwards, they are robustly transplantable. So we wanted to understand what is really this maturation process that gives them their functional properties. So we zoomed into those highly molecularly purified HSCs and looked at key programs that are either downregulated or upregulated during this maturation process. And we can see the downregulation of endothelial genes, proliferative genes, known fetal genes, and at the same time we can see upregulation of some key programs, most notably MHC class II molecules, such as HLA-DRA, here's an example, as well as PROM1 with CD133 that has been used as a stemness gene in many systems. And these really are upregulated at the stage when HSCs become transplantable, and because they are surface markers, we can witness their upregulation also as HSCs are maturing in the fetal liver and ultimately in the cord blood. And so now we have a little bit of a handle how to find the HSCs, how to define their maturation state, both inside and outside. And so we're beginning to have an understanding of this process that needs to happen before you can make a functional HSC. But what about the origin of HSCs? By using UMAP, we could connect our HSC cluster to endothelium, and specifically arterial type endothelium. And by using the key markers of hemogenic endothelium, which means you're still endothelium, you're turning on the HSC specifying factor RUNX1, but you haven't turned on yet the pan-hematopoietic marker PT-PRC, we can identify this hemogenic endothelium that is transitioning. And here shown in blue, we can see that it's highly HOXA patterned, which we had shown before is a key marker for the definitive hematopoietic fate. We can see that hemogenic endothelium is indeed part of arterial endothelium. There's been a lot of debate about that, but not just all arterial endothelium. There are specific genes that are highly expressed still in the hemogenic endothelium, but specifically in this population that we call the pre-hemogenic endothelium that may be preceding the HSC specification. And here I would like to highlight IL-33, LDH1A1, as markers of the pre-hemogenic endothelium, and KCNK17, another marker of hemogenic endothelium. So this we predicted from the RNA when we went to the human embryos and looked inside the dorsal aorta, where we see these hemogenic clusters that are the proposed sites of hematopoietic emergence. We can find exactly the transition from an IL-33 positive pre-hemogenic endothelium to upregulation of KCNK17, the marker of hemogenic endothelium, and ultimately SPINK2, which is marking those cells that have already transitioned to become hematopoietic. So this was very promising, and the reviewer said, how about this gene and that gene and that gene and this gene? And I said, we're not going to be able to stain all these as proteins, but we decided we're going to give it a shot and do Visium spatial transcriptomics, and we did, and we sliced the intact embryo to 7 slices and did this Visium, and we can here see, this is the dorsal aorta, we can see the expression of IL-33 in the right places, LDH1A1 on the ventral side as initiating retinoic acid signaling, which we now know is very important also functionally. KCNK17 is only turned on in those sections where we see visually an intra-aortic hematopoietic cluster, as well as SPINK2, which is then a marker of emerged HSC. So now we can go back to the same data set and look at other genes, what is unique about the environment that is specifying an HSC versus a different type of stroma, for example. But now I've bragged about all these markers, but how do we know that they are specific to the HSC wave versus the earlier progenitor wave? So we don't get access to these very early tissues, but by looking at data from Zheng et al. Paper from 3-week human embryo and comparing our 5-week human embryo hemogenic endothelium and HSCs to those very early ones, we can find that there are tissues that are unique that there are markers that are absolutely unique to the the definitive wave, such as here shown ADGRG6, for example, IL-33, LDH1A1, as well as some key HSC transcription factors that are not shared at all with the earlier wave. So if we now look, what have we learned? We've been able to define the HSCs as they're born in the AGM, define their endothelial precursor, follow them throughout human development, and find the key stages and markers for their maturation. So what can we now do with this? We of course want to the field to be able to utilize and now ask, what are the cells that everybody is making in vitro? How close are we to make the real HSC? And there's been a tremendous improvement in the field through following these developmental cues. It's been possible now to create these different waves of hematopoiesis. So we collaborate with Andrew Elephantis' group, who said, we think we've made an AGM like hemogenic endothelium, let's see what our cells look like. And we first used AI so that we could take completely hands off, and it said, yes, most of the cells that you're making really match to the AGM state HSCs, and we can see all the endothelial intermediate. We have some placental HSCs, but there's nothing that matches the fetal liver stage. And when we compared our HSC maturation scorecard, we could see that these pluripotent cell-derived cells really match more closely to these very nascent HSCs, and they don't upregulate these maturation markers such as PROM1 or HLA-DRA. So we really decided that this should be now the next goal, is how do we take our cells further into the, it's very promising in the footstep of making a self-reading HSC, but how could we make them functional all the way? So Mengwei Ko, who was wonderfully trained in the Dan Kampfan lab, is now came to our lab to take this challenge, and we've been working with Andrew Elephantis' team utilizing the protocol, a very sophisticated protocol that is very highly, defined stages of when different developmental cues are being applied. But rather than stopping right there, we're going to be continuing later. And we use HLF-reporter. We've showed before that HLF is the one marker, one transcription factor that is most specific to the undifferentiated HSCs. So we can see that there's no HLF when during early hemogenic endothelium specification, when we already see CD34 and CD90, but it comes expressed as HSCs are emerging, and we see very nice HLF positive populations that co-express all these key HSC markers. So now if we put them in the expansion culture, Mengwei showed that they are fully loaded initially. We can see this huge expansion of CD34 population, and even when we look at the HLF population. But are they still immature? We've went back to our gene expression data and looked at some of these markers such as ADGRG6, IL-3 receptor alpha, that are the markers of the more immature HSCs, and then PROM1, which is the marker of the maturing HSCs. And it's very exciting that we can see they're initially highly ADGRG6 positive, which gets downregulated as we are upregulating some PROM1 expression. So we think we are on the pathway of with also recapitulate HSC maturation in culture, but we're not sure yet if they are fully functional, but this is then for the next talk. But we felt that it's absolutely important that we really understand better of the mechanisms that maintain HSC function, as well as our key intrinsic regulators of this maturation process. So we went back and looked at turn back to our old friend MLT3, which we had found previously, because MLT3 expression always goes with the self-renewing cell. It always goes down when HSCs differentiate or when we put them in culture. It's highly enriched in HSC, some expression in erythroid lineage. It's part of the superelongation complex and DOT1L complex, so it can regulate transcription at the transcription elongation step, as well as maintain active chromatin marks. And we showed when we knocked down MLT3, this is already published, so I'm going very fast, that if you knock down MLT3, you lose all the undifferentiated cells in culture, and they don't transplant. So absolutely critical for HSC self-renewal. What about if we restore the expression that is being lost when we culture HSCs? We could see that when the control HSCs are running out of steam in a couple of weeks, if we maintain MLT3 expression, we can expand this immunophenotypic HSC population, and when we transplant, we could show that we could get at least over 12-fold expansion compared to the uncultured cells, and more than 5-fold expansion compared to the control cultured cells that uses the best conditions at that time with all these new small molecules, UM171, SR1. And they didn't transform. They were not lineage biased. They really behaved in vivo like real HSCs. We showed that MLT3 binds to key HSC transcription factors, and most likely through DOT1L dependent process is maintaining their expression as HSCs divide, really witnessing symmetric self-renewal in culture. Well, how do we know for sure the protein complexes which MLT3 works with its IP mass spec Iman-Fares in our lab and in cord blood HSCs in culture, and showed indeed the interaction with the superelongation complex and DOT1L complex, and as well as some other complexes that we're currently validating. But it was still fascinating for me, like, why don't we, if you overexpress MLT3, why don't you lock it into a self-renewing state? And that why do they still remain responsive to the microenvironmental cues? So we tried to understand this by looking into the regulatory elements for MLT3. And it was interesting when we look at the histone mark in fetal liver HSCs, we found the expected transcription start site. But when we looked at those histone marks, it looks like there's a second transcription start site. And if you looked very closely to some random browsers and things, we could find like there was some evidence maybe of a short isoform of MLT3 that never had been published or never reported. But we could see that this second TSS was rapidly closed when HSCs differentiate, and there was also an enhancer that behaved in a similar way. And if we look at the RNA-seq data, we have a peak here that corresponds to a potential unique exon here that is not in the canonical isoform. So this is predicted to form a protein that lacks the chromatin-binding Yeats domain, but still maintains these protein interaction domains. And this was fascinating for us because this is also the same portion of the gene that is part of the leukemia oncogene, the MLL-MLT3 fusion that can confer self-renewal to progenitors. So this was very interesting for us. We wanted to see if it's actually making a stable protein. We overexpressed MLT3 short, and we could see the protein of expected length. And as we predicted, because it's lacking the nuclear localization signal and Yeats domain, it's not going to the nucleus, it's not going to the chromatin. We see some in the nucleus, but mainly it's in the cytoplasm. But it was very interesting for us, especially when we started looking at the expression patterns more closely. And even from the short read data, we could see that this exon here, or this peak, could really only come from the short isoform. So we could see that when HSCs differentiate, they lose the short isoform even more rapidly than the long isoform, also verified by PCR. And when we put HSCs in culture and zoom into those that can still maintain an HSC phenotype that has some remnant long isoform left, but the short isoform is downregulated even more rapidly also in culture. So this made us very, very excited. So we said, what if we now express the short isoform? Maybe this is the isoform that expands the HSCs. Maybe we always express the wrong isoform. But this wasn't the case. There was absolutely no expansion when we expressed the short isoform. Only the long isoform expanded immunophenotypic HSCs when we expressed them together. The trend was the same, but it was suppressing the expansion by the long isoform. So we were like, okay, let's make sure that we are expressing the right thing. If you look very carefully, there's actually sometimes there's two peaks here. So we went back and we said, oh, interesting. There are maybe 2 different subvariants of the short isoform that may have 1 or 2 of these non-coding exons in the 5' UTVAR. We verified this by 5' raise, but when we overexpressed them, they made the same protein as predicted, but it was still a little bit puzzling. If you pull the ensemble today, there's 10 isoforms for MLT3, and we're like, oh, we thought that there are 2, there's 10. So we did PacBio. This is really data from last week. We're super excited, and we really were able to witness that everything that we kind of indirectly put together is true. The main isoform is the long isoform. We have 2 subvariants of the short isoform, and it's in fetal liver we have less of the short, and we have a little bit more of these 2 subvariants in the cord blood. But now we can at least be comfortable, 6 years later, that we have actually been studying isoforms that really are the main isoforms in real life. So what if we knock them down? We did isoform-specific knockdowns using lentiviral shRNA. And the long isoform, we already knew what to expect. You lose the undifferentiated HSCs. We actually see this premature appearance of 38 cells in culture, and then the premature loss of the undifferentiated HSCs. When we knocked down the short isoform using 2 different shRNAs, we actually didn't see a loss of HSCs. We saw the opposite. There was actually more of these EPCR-positive candidate HSCs initially. And we did knock out both isoforms. It was a hybrid phenotype, initially mimicking the short isoform loss, and then ultimately mimicking the long isoform loss. But so this culture gave us an idea that maybe these are doing something very different, these 2 different isoforms. But when we transplanted them to immunodeficient mice, the long isoform knockdowns didn't transplant at all, but also the short isoform knockdowns that look like we're expanding better in culture, they also were not able to engraft properly. So really showing that, that this is a critical that both isoforms are critical for HSCs. So quickly to look at their, what are these isoforms regulating, we showed that they, when we look at the programs, there are many key HSC programs such as protein synthesis and transcription that are really the targets of the long isoform, and they are regulated in opposing directions with the short isoform. Whereas we found programs that seem to be repressed by the long isoform, such as splicing and mitochondrial metabolism, that are going the opposite direction when we lose the short isoform. And here are individual examples, and we can see that they are direct targets of the long isoform, really showing that we are both looking at activating and repressive genes that are regulated in opposing directions. When we looked during development, we can see that MLT3 is expressed already in hemogenic endothelium, but there is no short isoform. It comes expressed later during development. So what is it doing during development? We quickly, I'm going to go through this data very quickly. We showed that also when we overexpress it at the fetal liver stage, we see this opposing regulation between the long and short isoforms. And we identified several interesting candidates, which I'm going to mention. This one gene, IGFPP2, it's a known fetal gene that is expressed in highly proliferative tissues, becomes reactivated in cancer, is already down in the fetal liver HSCs when they are mature, but gets reactivated in culture. When you put HSCs in culture, there's this rejuvenating signal, and the long isoform is boosting it, whereas the short isoform is repressing it. And we see the opposite when we look back into the knockdown data, suggesting that maybe these isoforms are involved in this shift from the highly proliferative proliferative fetal stage to a more homeostatic state. And when we knocked down IGF-BP2, we couldn't see any HSC expansion culture, and we showed that this was when we overexpress MLT3 and knocked down IGF-BP2, we didn't see the positive effects of MLT3, really showing that this is a one mechanism by which the MLT3 long boosts this highly proliferative, more fetal-like stage, whereas the MLT3 short is maybe enabling them to function more in a homeostatic manner. So final question, so why does the long isoform overexpression alone works if you need both of them? When you look at it, the long isoform is actually binding also to its own TSS as well as to the TSS of the short isoform. And we see this specifically when we are overexpressing the long isoform. So when you're overexpressing the long, you actually get the whole family to work together for the same price. And we think that this is really the reason why the overexpression of the long works, because you still have a foot in the door to maintain the expression of the short isoform, and this is the only way why we can have a fully functional transplantable HSCs. And so to summarize, we formed that there's not only a one, but two main isoforms of MLT3 that are critical for HSC development and maintenance. And the long isoform promotes specification and expansion. The short isoforms is one of the key factors that facilitates this transition to a more homeostatic state where HSCs can actually engraft. They're both very vulnerable in culture, but when you overexpress the long isoform, you not only restore the long isoform expression, but also keep the foot in the door for the short isoform being there so that HSCs can transplant. So I think about them as the gas and the brakes. You have a car, you don't just want to have a car that has gas, you need the brakes to know when to turn and turn safely. So I'm thinking about MLT3 not as a gene, but as I call it a stemness operating system. How about that? And where you can, these two isoforms are enabling HSCs to do these developmental transitions as well as responding, at least in part, reversible fashion to the key physiological cues. So boom, 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 boom. No time to show anything about that, but definitely want to show my acknowledgement slides. So many people to thank. I didn't know any of this. Vincenzo Calvanese started all the MLT3 studies in our lab. Anastasia Vavelina, who just defended her thesis, took over the short isoform project. Vincenzo and Sandra did the single-cell RNA-seq. Iman did the MLT3 mass spec, and many collaborators to thank for. Mengwei Ko is now working on our pluripotent cells, and of course the next question is what happens if we can restore fully both isoforms also during our pluripotent stem cell maturation. So that will be for the next one. So if there's any time, I would be happy to take questions.
Thanks, Hanna, for another terrific talk. When you alter the relative expression of the short and long isoform, do you affect overall translational output and translational activity? Have you measured that?
So, I know we've talked about with you like many times. We should really do the OP Pura experiment. We haven't done it yet. So right now, we would predict that it does, but also why we haven't done it, because I don't anymore think about that it's one target gene or one thing. Like, it's really, I think that both of these isoforms, they have the ability to modulate these key programs, not just the one, but I think that protein synthesis is going to be one key component, and we see it also that was in this developmental transition. You've shown very nicely that the fetal HSCs have much higher protein synthesis than the homeostatic ones. So we think that these isoforms really are, but that will be then hopefully for the next paper to really dissect in more detail.
And so we should definitely do the experiment. Yeah. But also, is the relative translational efficiency of each isoform different? Have you measured that at all? Oh. And is there does that create like a circuit and a feedback where it's regulated at the level of translation to control? Okay, so now you are asking, so translation control of the isoform. So here is, currently we cannot, we don't have an easy way to look at the native short
MLT3S. We are looking into generating pluripotent cell lines, where we could hopefully tag, if we can still maintain their intact function, that we could actually look at the native proteins and their protein levels in that sense. But currently, we don't have an easy way to really study that, but definitely interesting to think about.
I have an idea for that. Protein simple, we've got it in the lab. So you could do it by PI. The two short isoforms and the long probably have a different isoelectric focusing point. But my question was more about why you get the long versus short, because we see this splice isoform switching for other genes. Have you looked at MBNL1 expression, MBNL3, or RBFOX2? So there are certain genes, when you lose them, you knock them down, you revert to an embryonic transcriptional or embryonic splicing program, which is kind of what it looks like there with your MLLT3, and actually MBNL1 has been implicated in MLL rearranged leukemia, so slightly different, but I'm just wondering if this is what you're really looking at.
So very interesting, although these two major isoforms do come from two different transcription start sites, so we think that they are kind of, we have the long that is first activated, and then the short has its own TSS, its own regulatory also enhancers, which I didn't show yet. They are already primed before it's on, so the hemogenic endothelium, they already know that they want to turn it on, but there's some different trigger that turns it on. But speaking of splicing, there also, we do see these two different subvariants of the short that don't change the protein, but may change something else for the RNA. So there is also alternative splicing that is happening. And we have some evidence that there may be some more rare variants, at least during development, where there may be some exon skipping that is happening, that they may modulate the function. So we need to look at now in the adult and aging and mobilization and all these situations, like if these subvariants become more common, but definitely want to look into splicing and RNA processing. So let's talk more.
So I'm trying to remember, which is the fusion protein? Is it MLL-T3, ENL?
So the fusion protein where MLT3, also known as AF9, is involved, so it's MLL, MLT3. So MLL binds chromatin, and MLT3 brings these protein partners.
Yeah, so those cells are malignant at the monocyte lineage, not at the HSC lineage, even if we put it into HSC. So you think it is the partner that changes that state at which it can be transformed? So
I think there's a few things, the reviewers were very like, are you sure you're not getting leukemia? Are you sure you're not I'm sorry, I don't get leukemia. But also, it only goes to the HSC genes if they are already, the TSS is open. So if you put it to a cell where those HSC regulatory genes are not open, it's not a pioneer factor. It's only a maintenance guy. So that's one important thing. Whereas when the fusion gene MLL1 is going and making its own rule where to bind and what to bind. So that's one thing. The other thing about leukemia in HSCs, HSCs express the short isoform also. So if it can give the breaks to the long isoform, can it also give breaks to the leukemia fusion protein? And this is one of the things we want to be testing now in the future.
I think we should move on. So thanks so much. That's wonderful.